I'm going to explain to you the demonstration of precipitin reaction. The precipitin reaction is nothing but the reaction between the soluble antigen and the soluble antibody. The soluble antigen is called as precipitinogen. Instead of antigen, it is called as precipitinogen, and the antibody is called as precipitin. Altogether, the reaction of soluble antigen with soluble antibody results in formation of precipitate, which is visible as white precipitate. And this white precipitate can be visualized in an inert gel, that is agarose gel. This was first demonstrated by a immunodiffusion technique by Ouchterlony, and this person was responsible for demonstrating the antigen antibody reaction by diffusion in the agarose gel. Hence, it is named after him as Ouchterlony technique. The soluble antigen and soluble antibody are made to diffuse towards each other, and at the point of meeting between the antibody and the antigen, a precipitin line will be formed. A thin line of white precipitin band will be formed, and therefore this reaction is called as precipitin reaction. Let us see what are the equipments that you require to demonstrate this simple immunodiffusion technique, where the antigen and antibody are made to diffuse in the agarose gel and meet at a point. You will require the microscope slides, you will require tissue paper and ethanol is used by moistening the tissue paper to clean those microscope slides. The surface of the microscope slide should be free from fingerprint. It can be cleaned by wiping the ethanol on its surface. Once it is clean, the fingerprint should not come on it. Then those slides are laid horizontally on a uniform surface. Okay. Then you need pasture pipettes to pour the gel. Each pasture pipette could take about 3.5 ml of agarose gel, and per microscope slide you would require about 3.5 ml of agarose. And you will need a rubber punch. The rubber punch is made up of a rubber bulb with a steel bore or an improvised device where a micropipette tip can be attached to that rubber bulb, and the tip of the micropipette tip is cut so that you will get almost about 3 millimeter diameter when you use it to punch the agarose gel. Okay, and this punch is used for punching the wells on the agarose gel, and these wells are used for loading the antigen and the antibody. And you will need the pre-marked stencil, and that stencil will have markings which can be seen through the transparent agarose gel, and based on the position of these markings one can punch the agarose gel so that you will get a uniform pattern. And a micropipette for loading the sample. The samples can be loaded into the well, that is antigen and the antibody into the well using a micropipette. You may require a pipette, and the pipette can be used also for loading the agarose gel on the microscope slide. And sometimes when you punch the wells, the agarose may not come out properly. In that case, you will need a needle to remove that agarose, which will be demonstrated later. Agarose of very high quality, which can dissolve in a suitable buffer should be used, and you will require the antibody, which is called as antiserum, and you will require antigen, and antigen in the soluble form and the antibody which is there in the soluble form will be used for the demonstration of the precipitation reaction. Initially, you take the required quantity of agarose, that is 2% agarose will be required, and that 2% agarose should be taken in suitable quantity of the buffer, and then that has to be heated on a hot plate at a very low heat. If it is a stove, you heat it at a very low heat. Either you use a water bath as a precautionary measure and keep in the boiling water bath the agarose that is mixed in the buffer. As the water starts boiling, the agarose will start melting. And once it melts completely, there will be no particles in the buffer and it will be transparent solution. The 2% agarose solution prepared in this manner, now it is ready to be used for the preparation of slides. See that there are no particles in the agarose that is dissolved in the buffer. Here I am going to demonstrate about the preparation of the slide with the molten agarose. I am taking one full partial pipette of molten agarose, which is 2%, in buffer and dropping it on the slides which are cleaned previously with alcohol, and by gently dropping and without disturbing the surface and by keeping the slides on a perfectly horizontal surface, the agarose will form a uniform layer over the slide. And 3.5 ml of agarose on the slide will form a thickness of 3 millimeter thick gel once the agarose solidifies. The slides prepared in this manner are kept in the laboratory temperature for 5 to 10 minutes until the agarose solidifies. Once the agarose is solidified, the slides are now supposed to be transferred into a moist chamber. And the moist chamber is nothing but a simple lunch box, plastic box, in which you prepare a bedding of filter paper and spread it uniformly. After spreading the bedding of filter paper, moisten that filter paper with distilled water. Moisten it sufficiently, but do not flood it with distilled water. Once the filter paper gets moist, once again press it hard onto the base of the box. The moist chamber is ready. The moist chamber prepared like this is generally used for keeping the agarose slides. Next, transfer the slides to the moist chamber gently without disturbing the surface of the agarose. It is already solidified, but handle it gently. After transferring the slides to the moist chamber, cover it with the lid and keep this moist chamber in the fridge, but not in the freezer. In the fridge, in the lower chamber, you keep it for about half an hour. This facilitates uniform distribution of ions which were there in the buffer, and these ions will distribute evenly in the agarose. In addition, by keeping the chamber in the cold condition, the agarose will harden and it becomes more solidified. Therefore, it will be easier to handle it because when punching, it will not crack. Next, you keep the agarose slides on the stencil. Here, the 3-well pattern is prepared. There are only 3 wells to be punched. 2 wells are going to be dedicated for antigen, 1 well for the antibody. The punching of the wells, whether it is 3-well pattern or 2-well pattern or 5-well pattern or 7-well pattern, depends on the number of antigens that you have to analyze against a given antibody. In the center, the rubber bulb with the steel tube is shown. Now we are using an improvised punch that is made up of rubber bulb with attached micropipette tip, and that micropipette tip is cut at the tip so that you will get approximately 3 millimeter diameter wells. By gently pressing the rubber bulb, press the tip to the agarose and suck off the agarose. So, in the next slide, I am showing punching of 7-well pattern. Here, there is a central well surrounded by 6 wells. That means the central well will be dedicated for the antibody and surrounding 6 wells will be dedicated for the antigen. So, 5-well pattern will help in analyzing 4 antigens against 1 antibody well in the center. 7-well pattern will help in identifying 6 antigen samples against 1 central antibody well. So same way, after punching the agarose, by gently pressing the tip of the punch onto the agarose, you can suck off the agarose. It will create the wells of about 3 millimeter diameter. Here, once again, I am showing the punching of the 7-well pattern. The slide is taken out from the stencil. Now, as I had told you earlier, if at all the punch has not created the well, you can remove the agarose from the well using the sharp needle without disturbing the agarose. But you need practice to do that. You need to be very gentle, removing the agarose from the well. You can see in the photograph now, in the video, how the agarose is removed. If a sucking pressure of the rubber bulb is not sufficient, this is how you will get the wells created, but agarose does not get sucked. Therefore, you must exhibit or exert sufficient amount of pressure using the rubber bulb. So the wells created like this now are ready to be used for the immunodiffusion, that is Ouchterlony technique. The central well will be dedicated for the antibody and the peripheral wells will be dedicated for the antigen to be loaded. And in each well, you can load about 10 microliter of the antigen or the antibody. Here in this diagram, it is already shown that central well dedicated for the antibody and peripheral wells for the antigen. Sometimes, to be on the safer side, you can dedicate one antigen well as a control well against the known antibody, because when the precipitation reaction takes place, it is 100% sure that the control well where the known antigen is added against the antibody will definitely form the precipitation band, whereas the unknown test samples which are loaded, if they are negative for the antigen against the antibody loaded there in the well, they will not produce the precipitation band. If you are dedicated all the wells for the antigens, then if no precipitation band appears, you may start wondering whether the reaction took place or not. Therefore, to avoid that ambiguity, one can load known concentration of antigen and corresponding antibody can be loaded in the central well, and that will definitely give the precipitation band. As I had told you, you have the antigen and the antibody samples ready with you. Now, those antigen antibody samples have to be loaded into the well using the micropipette tip. As I had told you that 10 microliter of sample will be sufficient to be loaded in each well of about 3 millimeter depth. And please see to it that the loaded sample does not overflow over the well. Overflowing will spoil the test. And gently touch the tip of the micropipette tip to the corner of the well and load the sample. And see to it that the tip is supported with your left hand when you are loading. Otherwise, due to vibration or gentle shaking of your hand that is holding the tip, may tear the gel. As a precaution, see how I am supporting the tip with my index finger of the left hand, and right hand I am using to hold the pipette. And by supporting the tip with the left hand, you can prevent that pipette tip being shaken. If you do not support the tip with your left hand, once again you see it clearly, there is a possibility that when you are touching the corner of the well, due to little bit of shaking of your right hand, the well may tear and that well becomes useless. Therefore, please see to it that whenever you are preparing the Agarose slides, the torn wells should not be used for testing, because that will give wrong results. Each time, see to it that for different antigen samples, you change the tip, because there is a possibility that some samples may be positive for the antigen, some samples may be negative. In that case, if you have used single tip, then it may contaminate and give wrong results. Therefore, for loading of each sample, you must use single tip and discard the tip. Now I am loading antibody in the central well. The slide is now loaded. In the peripheral wells, antigen was loaded. In the central well, antibody corresponding to test those particular antigens has been loaded. The slide has to be kept back now in the moist chamber. Remove the slide gently by lifting it from the stencil without disturbing the agarose. Keep in the moist chamber. Cover it up with a lid and keep it back in the fridge. Once again, not in the freezer. It should be in the bottom portion of the fridge. And leave it alone overnight. Next day, come and observe the slide. When you come and observe the slide, you will see that the slide will have developed the precipitin bands like this. As seen in the picture here, the precipitin bands can be seen between the antibody well and the antigen well. In this picture, you can see that this antigen well and this antigen well is positive for this antibody. So therefore, you have got a very good precipitin band. And this is thicker and the precipitin band is brighter compared to these precipitin bands on this side. This antigen is also positive, but amount or the concentration is comparatively lower compared to these two antigens. You can make out here, due to high concentration of the antigen and antibody, the precipitin band is brighter here compared to this side. Then when you come to these middle and the left hand side well, you can see that these precipitin bands are much lighter compared to the precipitin band you have seen on the extreme right side. That clearly indicates that even though these antigens are positive against the antibody, and these test samples have low concentration of antigen corresponding to that antibody compared to the antigens that are there here in the extreme right hand side, which is showing a bright thick precipitin band. Based on these precipitin bands, you can classify this precipitin reaction, and that classification is as follows. The precipitin bands, if they produce a smooth curve like this, the antigen-antibody reaction produces a precipitin band of smooth curve like this as shown here or in this diagram, where the antigen loaded is almost the same in both the wells. The epitopes of those antigens are the same. Epitopes or the antigenic determiners are almost the same, corresponding to the antibody that is loaded in the central well. Because of that, the diffusion and if the concentration of these two antigens are also same, not only the epitopes, their concentration is also same, they will produce the precipitin band right in the middle between the two wells. Now here in this diagram, you can make out the precipitin band between antigen well and the antibody well is almost in the middle. And these two precipitin bands have met at this point. And if you go to the previous slide also, you can see the precipitin bands have met and it is a perfect inverted V shape and a smooth curve like as we are seeing here. It's a smooth curve. The reason being these two precipitin bands are met. The reason being the antigens are of the same type. The concentration of these two antigens are also same, and the diffusion force exhibited by these antigens towards the antibody, which is also diffusing in all directions here, and the antigen is diffusing in all directions. Radially, it is diffusing in the agarose gel in all directions. And when they meet at a particular point, being equal in concentration, they will produce a precipitin band right in the middle between the two wells, and those precipitin bands, because of the equal diffusion force, will meet at a particular point to give you a smooth appearance like this. Therefore, it is called as line of identity. That indicates both the antigens are of same type, which are tested. Now coming to the second slide, that is line of non-identity. They are non-identical antigens as represented here. In one well, antigen A is present. In another well, antigen B is present. Correspondingly, the antibody A and antibody B is present in the antibody well. When the precipitin band appears between these two wells, these two precipitin bands, because of the dissimilarity of the antigens, will crisscross with each other as shown here. There is crisscross. It is not smooth as shown in the left hand side. The reason being the diffusion force exhibited by both the antigens, one. Second, they are different in nature. Their epitopes are different. Their determiners are different corresponding to that antibody. Because they are not identical to each other, they will show crisscrossing like this. This will clearly indicate that the antigens that are being tested are dissimilar in nature. Therefore, it is called as line of non-identity corresponding to that particular antibody. They will have crisscross. Now coming to the third type, line of partial identity, where the antigens which are loaded in the antigen well, some may be similar in nature and few may be not similar in nature. In this case, what has happened is, in the left hand side well, there is antigen A, and in the right hand side well, there is antigen A as well as B, which is not present in the left hand side well. Correspondingly, there is antibody in the central well with respect to these two antigens. When the reaction takes place, precipitation reaction takes place, you will see that as far as antigen A is concerned, which is similar, you will get line of identity like as we had seen earlier in the left hand side here. That is line of identity as far as antigen A is concerned. Whereas the another line that has appeared here, that is corresponding to antigen B with respect to antibody B, and it doesn't have a similar antigen in the other well. Therefore, that precipitation line has formed a spur which is extending out. There is no equal diffusion force on the other side, therefore it has not bent. So it has formed a spur. And that spur formation indicates that that antigen is different from the one which has formed the line of identity. The spur formation indicates that that antigen is different, and therefore it has remained separate as a precipitation line. Only thing is, because the concentration of antigen B and A is almost the same, therefore the precipitation bands have overlapped with one another. Imagine if the concentration of A and B were different, then the precipitation band of B would have come separately without overlapping with the precipitation band of A. If the concentration of B was less compared to A, the precipitation band would have appeared closer to the antigen well, and the precipitation band would have been here near the antigen well. And if the concentration of B was more than the concentration of A, then the precipitation band would have appeared closer to the antibody well, and it would have appeared near to the antibody well and crossed the precipitation band like this here, crossed. If the concentration of B was more, antigen B, it would have pushed the precipitation band towards the antibody side and it would have appeared here and crossed like this. And A antigen precipitation band would have appeared smooth like this, crossing like this. Or if the concentration of B was less, it would have appeared very close to the antigen. So that also shows that depending on the concentration of antigen and the antibody, if the concentration of antigen and antibody are equal, then the precipitation band will be right in the middle between the two wells. But if the concentration of one of them is higher, the higher concentration substance will push the precipitation band towards the lower concentration. Say for example, if the concentration of antigen A is higher or B is higher, it will push the precipitation band towards the antibody side. Vice versa, if the concentration of antibody B is higher corresponding to antigen B, it would push the precipitation band towards the antigen. Therefore, one need to be very careful while demonstrating the precipitation bands. The concentration of antigen and antibody should be approximately equal to each other, and if it is disproportionately higher in one, then it will form a soluble complex and there will be no precipitation band appearing. Therefore, precaution is very important. To demonstrate the pristine band, the concentration of antigen and antibody should be approximately equal. They need not be same. The antigens need not be same which are being tested. Based on their nature, they can be identified using the pattern of the pristine band. One can see whether the antigens are related by the line of identity if it is a smooth curve. By the line of non-identity, it shows that those antigens are not related to each other. They are different, their epitopes are different, their determinants are different corresponding to the antibody. And if they have partial relationship, somewhat similar but slightly different, you can see the spur formation and that will give partial identity. So this is how you can demonstrate a precipitation technique or immunoprecipitation reaction by a simple immunodiffusion method, which is called Ouchterlony immunodiffusion method, by making the soluble antigen and the antibody to diffuse across each other in the inert agarose gel and meet at a particular point to produce a precipitation band. Thank you.